Welcome to LFPL's At the Library series, an ongoing podcast featuring author talks, programs, and events at the Louisville Free Public Library. For more information about upcoming events, visit us online at www.lfpl.org/upcoming events.
Please welcome Dr. Lee Dugatkin.
Thank you. Thank you. Thanks again, Tony. So I have to say, I really enjoyed last week, and I'm glad to see that there are a lot of people that came back. And I'll say my favorite part by far was the question and answer session. Now, I never said this, but the questions were way better than the questions I get from my students at the university. And absolutely on par with the questions I get from when I go speaking all over the place. So it was great. I hope that people will ask lots of questions again this week. So as I mentioned last week, we're going to meet for 4 times, and last week was sort of this just a general introduction in terms of evolution and natural selection. This week, I'm going to focus on something a little bit more specific, which is the question of how do we get new species and how do we lose species? So we know of millions and millions of species that exist now, and that's probably just a fraction of the ones that are out there. Most species that are out there are likely undocumented. And so evolutionary biologists, ever since Darwin, have been interested in this process of, well, how do you get species and how do you lose them? And those are the topics of speciation, the formation of new species, and extinction. But it turns out that before you can really discuss those topics, you need to step back and think about what a species is. So at some level, we all know when we're outside that robins are different from squirrels, and they're different, and those are different from dogs, and those are different from bullfrogs. But it turns out that if you think about it on a very grand scale, it's not so easy to know immediately what's part of this species versus what's part of that species. So I think the example I briefly mentioned yesterday is there are lots and lots of different species of squirrel. Right? And some of them look a great deal alike. How can we delineate this is part of one species, this is part of another species? And that would hold true not just for mammals like squirrels, but for almost everything that's out there. There are some very clear differences, but there's lots of gray zones, lots of ambiguity, where it's not so clear that this is the same species as that, or it's not. And so the first thing that evolutionary biologists do is they dive into this question. Okay, so how are we going to define a species? And I'm going to start with an example that's more meant just to get you thinking about that question. So what you're looking at there is an empty subway, an empty tube station in London. And back during World War II, the Brits would live at times weeks on end down here when the Blitzkrieg was going on and London was being bombed. It was one of the very few places that were safe. And once people survived that and came back up, they began talking, and they talked about all sorts of things. And one of the things they talked about was that the mosquitoes down there seemed really different than the mosquitoes above ground. They seemed way nastier, way more likely to bite humans, and they just seemed to be different in every possible way from the mosquitoes that were just above ground at that spot. And in the late 1990s, evolutionary biologists went and they studied populations of Culex mosquitoes that lived down in the tubes and above ground. So the mosquitoes have been down there probably since the tube was founded, which I can't remember now, but it's way more than 100 years. And so there's plenty of time. That's a lot of mosquito generations. There's plenty of time for evolutionary change to happen down there. And as we'll see, there's some evidence that that has gone on, and this is going to allow us to sort of conceptualize, well, how can we say whether or not something's this species or that species? So let's take a look at what people found when they looked at mosquitoes above ground and below ground. All right, so Culex pipiens is just the scientific name for this species of mosquitoes that you would see above ground. You will note here that underground, so C. pipiens, that's Culex pipiens, is above ground. Underground, biologists have given it a subspecies name. In other words, something that distinguishes it from the above ground. And it's Culex pipien molestus, basically because that's what they do, is they molest us down there, okay? Now, when you look at the above ground and below ground varieties of this mosquito, right, you find all sorts of different things. So they're breeding in different places, either above ground or below ground. But here's an interesting thing. If you give them the choice, and so the researchers would grab mosquitoes from both places, they bring them into the laboratory, did a bunch of stuff with them, right? And so if you give them the choice, the above ground mosquito prefers to mate in open spaces. The below ground, in the tube station mosquitoes, prefer to mate in more confined spaces. So where they want to mate has changed over the course of many, many mosquito generations. I said that they're really nasty, they bite mammals, and particularly they like to bite humans. Okay? Above ground, C. pipiens mostly specializes on birds, right? But there aren't many birds down, there are some, but there aren't many birds down in the tube stations. And there, if they have the choice, again, you bring them into the laboratory, you give them the choice, they prefer to bite mammals, whereas the above ground variety prefers to bite birds. Now this one, egg production, might not seem as dramatic, but it really is if you kind of take a mosquito eye view of what's going on here. Above ground, the mosquitoes need a blood meal in order to lay eggs. But below ground, they don't. They will lay eggs right after not, in fact, getting a blood meal. A huge difference just in terms of what's known as their life history, sort of how we can describe them. And then there's this. Above ground, the mosquitoes go dormant in the winter. But below ground, they stay active all year. Below ground, the temperature is much more consistently warm all year round, whereas above ground, that's not the case. That's a huge difference in these varieties. Some of them basically go dormant. They go to sleep, they die. Some of them, many of them die in the winter. These guys are active all year. Okay. But for an evolutionary biologist, what's most striking about the difference between the above ground and the below ground varieties of this mosquito is that there is no mating going on between the above ground and the below ground populations. What's more, if you bring them into the laboratory and you try and mate the mosquitoes, if you bring in two above ground, they're happy and they mate. You bring in two below ground, put them together, they mate. If you put the above ground and the below ground together and there's nothing else around, eventually they will mate, but they will produce no viable offspring. Okay? No viable offspring. For an evolutionary biologist, this is a huge thing. It basically means there's no exchange of genes between the above ground and the below ground. And not simply because physically they're above ground and they're below ground, even if you put them together after many, many generations, so 100 years plus in mosquito generations in the tube system, now they produce no viable offspring. So we're going to kind of return to that notion of mating and producing viable offspring. But here all I want to get across is this question. So are they different species or aren't they? Is all of this, plus the fact that they never produce any viable offspring, is that enough of a reason for us to call them different species? If I just pulled a mosquito from below ground and above ground and showed them to you, and you didn't know this chart, there's no chance in the world that you would think they were different. They look very, very similar, but all these things are in place. Is this enough? Isn't it enough? Would it be enough if only this was happening, but they could mate and produce offspring? Or do they not have to be able to produce offspring for us to consider them different species? This is to get you thinking about those questions. So if you look, there are a number of what are typically referred to as species concepts, ideas for how it is that we can delineate one species from another. And I'm going to talk to you about three of those ideas. One of those is known as the typological species concept. Okay? This actually can be traced back to Plato. And Plato had this concept of essence, that things had an essence. And not only living things, but inanimate things had an essence that you could use to define them. Tables had a certain essence that made them tables and not chairs and not walls. So everything had an essence, and it was that essence that made it different from other things. In terms of defining species, early on, natural historians, and I mean sort of the 1600s, the 1700s, even into the 1800s, used this kind of thinking to define how we can break up the world into different species. Things that have the same essence, they're part of the same species. Things that have different essences, they're not. Of course, right? The question is how one defines essence. But essentially what this typological species does is it thinks of all the variation that you might see, all the different kinds of things that you might see when you're comparing this group to that group, as noise. All that matters is the essence. And if they have that essence, they're part of the same species. And if they don't, they don't. They're not part of the same species. All the naturally occurring variation, that's just noise. You don't have to worry about that. The question, though, is how do you define an essence? And again, we're talking about this concept in the 1700s mostly, even 1600s, up through the 1800s. And generally what you want to do is pick some kind of critical measure, something that's so bloody obvious that it's the essence of that organism that there's no argument. Now, as you might guess, that turns out to be fraught with endless problems. Let me give you a couple of examples. So this is a rat snake. Okay? They're all over the United States. I think they're in Kentucky too. Okay? So if you were a typologist in the 1700s, if I'm a typologist in the 1700s, what I say is what defines this species of snake is the orange and the kind of patterning that you see here. I mean, that's the most obvious thing about that a lot of other snakes don't have. Right? And so maybe that's its essence. This is a rat snake from a different place in the southeastern United States. It's yellow and it has black stripes going down it. If I'm a typologist in the 1700s and this is what I see, I'm likely to say, well, there are all these snakes out there. The essence of what I'm going to call a rat snake is it's yellow and it has these dark stripes. These are rat snakes from another population, not all that far away from those two populations. And in fact, here's a map of the distribution of populations of these colored rat snakes. Any trait that you pick and define it as the essence is going to have this problem. There's going to be real variation out there that you simply cannot ignore as much as you want to as a typologist.
These all interbreed?
Yeah, absolutely, absolutely. Here's a red fox. If I'm a typologist, it's red. And that's what and this is what a red fox looks like. Turns out that there are varieties of red fox that look like everything you see up here, including the silver fox, which, if I had time, I would talk about it endlessly. But these are all varieties of red fox. And any trait you picked in red fox, not just its color, would cause you the same kind of problem. So people pretty much around the time of Darwin began to abandon this typological species. But what they did was they abandoned it in its strictest form, but they started developing another version of the same sort of thing that's known as the phonetic species concept. And in a second, I'll tell you about the word phonetic. Okay? But as a general observation, organisms in the same species have similar what are known as phenotypes. And a phenotype at the level of an individual is just the suite of all the different things that you can measure about it. So you measure morphological things, bone length and bone density or whatever it might be. You measure anatomical things, you measure developmental traits, you measure behavioral traits. And things in the same species tend to have the same phenotype. Okay? So from that phenotype comes this phonetic species concept. And essentially, here's what the phonetic species concept does. It says, obviously, we can't go with this. This essence idea is not going to work. We can't pick one thing that defines a species, right? But we can measure lots and lots and lots of things about organisms and see whether or not things cluster together. So here is some phenotypic trait. Let's think of it as bone length. Okay? Here is another phenotypic trait 2, and let's think of it as fur coloration, or whatever you like. Okay? And we go out and we measure a bunch of individuals, and here's what we see. Some here, some here, some here. In other words, some have really thick bones and this color, some have really thin bones in this color, some have really thin bones and that color. What the phonetic species concept says is we're going to draw circles around those clusters, and we're going to call them species. So what it's doing is instead of picking one trait here, the essence, in this case, it's using 2 traits and saying things that contain those 2 traits we're going to call a species. But in fact, this is the simplest version of the phenotypic species concept. It's 2 traits that I'm showing you on a graph. You can do the same thing for 3 traits and see whether or not there are clusters of individuals who have one variety of those 3 traits and others who have different. And I'll show you a graph of that in a moment. You can do it for 4 traits, except I can't think very well in 4-dimensional space, so I can't show it to you on a graph. And you can do it on 12 traits, but I can't present it in a 12-dimensional analysis either. But computers can. Computers can cluster any number of traits the way that we've done here. And so what phonetic species proponents do is they collect data on lots and lots of traits: bone density, fur length, body length, beak size, hind limb size, any number of different things, and then they use sophisticated statistical software to look for the equivalent of these clusters, things that share phenotypic trait 1, 2, 3, 4, 5, 6, 7, and 8. And another group that share a different version of the phenotypic traits, except instead of 2, many, many, many. So it's still looking for similarity in traits. It's just not focusing on one trait and calling it the essence. So let me give you an example of one set of species that this has been done on. So these are medicago shrubs that you're looking at here, A, B, and C. And people who study these plants for a long time argued that those 3 different kinds of medicago shrubs are different species. But of course, what we want is some way to measure that. And the phenotypic species concept gives us the ability to do that. So what researchers did was they went to lots of populations of each of these varieties, maybe species, of medicago shrubs. And they collected them, and they measured everything they could about them. This analysis is statistically complicated, but these axes, principal component 1, just think of that as one cluster of traits that you could measure about these plants. And phenotypic, principal component 2 is another set of traits, a cluster of traits you can measure. And same thing for phenotypic, for principal component 3. Basically, they're measuring dozens and dozens of different characteristics. And they're plotting it out in 3 dimensions. And when you do that, what you find is that of the 7 populations that they went out and sampled from, of the 9 populations that they went out and sampled from, 7 of them clustered together over here. And those were what were called the arborea populations. It's what people thought was the arborea species. Now they have some data to suggest that it is, because everything that was collected from these 7 populations clustered together, and these 7 populations were, in fact, the populations of this arborea version. And it's separated out from the one population that you see in green and the other population that you see in blue. And those represent 2 different species. So all I want to get across here is that the researchers thought these 3 things were different species, but they had no way of documenting it until they measured tons and tons of things about them. And when they did, they found this cluster that represents one species, and these points that represent the other species. So this is obviously much more prevalent than the other 2 species, but these things are clearly different on a composite measure of all the stuff that they studied in the plant. People still use the phonetic species concept today, and often. It's particularly useful in paleontology when you're studying fossils, when all you have are bones and you can measure all sorts of aspects of the fossil. So it's typically used in paleontology, and it's actually often used to study microbial species, bacteria, viruses, other single-celled species. And I'll kind of come back to why that is a bit later, but this is where you often see these phonetic species concepts being applied. But there are problems with the phonetic species concept, and there are kind of on-the-ground practical problems, and there are conceptual problems. And I just want to touch on a few of them. So in terms of practical problems, over and over, the question arises whether or not some traits provide you more information that's valuable in delineating species than others. And so you'll have a naturalist who's studying some group of species and saying, I've been studying these guys for 40 years, and I'm telling you, this is what's really important. And another group saying, no, I've been studying it for a long time, and in my populations, this is what's important. And this is a practical problem. There's no solution to that problem. Are some traits more important than others? It's going to be a subjective decision. If some traits are more valuable than others, then what that means is that what you do in your statistical analysis is you weigh those traits more heavily. In other words, when you're doing your statistical analysis, if you believe that trait 1 is really what matters the most, more than 2, 3, and 4, then you set it up in your computer simulation that has the largest effect on the outcome. But if you think trait 2 and 3 are really the ones that are most important, then you weigh those more heavily. This is a problem, and it doesn't go away. There's also the question whether or not more traits are always better than fewer traits. Maybe some people say yes. The more traits you can measure, the more accurate. Other people say no, not necessarily. There might be some traits that are just much more informative than other traits. And I might not have as many as this research lab over here does, but the ones I'm measuring are really the most informative. And so if I have fewer of them, that's fine. That problem doesn't go away. And there are other on-the-ground practical problems. But from an evolutionary perspective, it's the conceptual problem here that's most daunting. And that is that this phenetic species concept assumes that organisms that are phenotypically similar to each other are evolutionarily related. In other words, if I do my analysis and I get these clusters and I say, okay, this is species 1, and this is species 2, and this is species 3, right? I'm saying that my 3 species of squirrels are more closely related evolutionarily than this species of squirrel and a dolphin, or this species of squirrel and a frog. They're evolutionarily closer. And I'm making that statement because I'm assuming the more phenotypically similar they are, the more likely we're talking about evolutionary relatedness. But you can have unrelated groups of organisms that look alike, not because they have some ancestor, evolutionary ancestor, that links them together, not because they're evolutionarily related, but because they've responded to the same natural selection pressures. And that's why they look alike. So imagine that and this phenomenon that we're talking about, the fact that unrelated organisms can look similar if they've been under the same natural selection pressure, is called convergent evolution. Let me give you an example. Suppose that you are a scientist who's come here from another planet and you don't know anything about life here, and you're going to apply the phenetic species concept. Because it's kind of easy. You can just measure stuff and look for things that cluster together. Right? Here are 3 things that on the surface, again, remember, you don't know what a shark is, and you don't know what a dolphin is, and you don't know what an ichthyosaur is. Right? They all kind of look relatively similar. But one's a fish, one's a mammal, and one's a reptile. Why do they look this way? Why is it that phenotypically they would cluster together? Why? Because they've all have to deal with being a large creature swimming around underwater. And there are certain things, body shape and other things, that work underwater. And natural selection is going to favor them whether or not you're a mammal or a reptile or a fish.
It didn't work for the dinosaurs.
So it didn't work for the dinosaurs? I'm sorry, meaning?
They were too big.
Oh, right. I'm not saying necessarily that being big is a good solution. I just want to say that they all, these 3 things kind of look alike, and so if you were just measuring stuff about them, they might cluster together. That's a big problem. Because of that problem, the most widely used definition of a species comes from what's known as the biological species concept. So this is the second species concept we're going to talk about. And here, a species is defined as groups of actually or potentially interbreeding populations which are reproductively isolated from other such groups. So here, the key is going to be whether or not you have mating going on. If things will mate with one another, but they're reproductively isolated, meaning they won't mate and produce offspring with other things, then they're part of the same species. But if they will mate with something different, then that thing isn't different if they'll mate together. If they're reproductively isolated, if you get mating within something, but not between that something and other things, then that something is a species. So what this does is it looks directly to an evolutionary mechanism, mating, to define what a species is. If individuals in one population can mate with individuals in another population, then they're part of the same species. And we say that they share the same gene pool. In other words, by mating, they're swapping genes with each other all the time, right? Because they're sexually reproducing organisms we're talking about here. And so they mate. You get offspring that have genes from one and genes from one parent and gene from another parent, right? So if individuals in two populations will mate with each other, then they're part of the same species. But if populations are reproductively isolated, right, then individuals in those populations are considered to be part of the same species and different from individuals in another population that they won't mate with. We can show this schematically. Imagine that we have what we're calling here 6 populations. And these lines mean gene flow. In other words, there's mating going on between 5 and 2, and 2 and 3, and 3 and 5, and 1 and 3, meaning individuals from here will mate with each other, they'll mate with these individuals, they'll mate with these individuals, and so on, right? And individuals in 4 and 6 will mate with each other, and then we see there are some other populations where there is much less gene flow between that population and anything else. So if we're going to start delineating species, then what we would say is these populations up here, where there's gene flow between the populations, they make up one species. Population 7 does not have gene flow with anything else. And so it alone is a species. And species 3 is made up of population 4 and 6 because there's gene flow between them. So the key is mating. Mating leads to gene flow. If there's gene flow in mating, then things are part of the same species. If there isn't, they're part of different species. And because reproductive isolation is so important to this biological species concept, people have looked for ways that populations become reproductively isolated, ways in which you don't get mating between individuals in 2 different populations or 10 different populations, whatever it might be. They've looked for mechanisms that might make that possible. One of those mechanisms is ecological or habitat isolation. You might have 2 populations of the same kind of organism, but if they're miles apart and they don't travel miles, then they can become reproductively isolated simply by distance and ecology. If one population prefers a grassy habitat and the other one prefers a more open savanna-like, a more open habitat, right, then you can get a buildup of reproductive isolation. They're just not going to interact with each other and you're not going to see mating. So you can basically separate them out that way. And that's sort of the easiest kind of mechanism. There's also seasonal or time isolation. So imagine that you have 2 populations of things that look very, very similar to you, and you want to know if they're different species. One way you might get reproductive isolation building up between those 2 populations is that in one population they breed in the spring, and the other population they breed in the fall. Otherwise, everything's the same. But they don't mate with each other then, because there's this isolation by season or some other aspect of time. Maybe in one population they breed in the morning and in another population they breed at night. So this is another way you can get buildup of reproductive isolation, of individuals in one population not mating with those in the other and becoming, therefore, two different species. You can get behavioral isolation. Just trying to think about how much I could. Okay, so I didn't know if I heard someone say something. So a good example of behavioral isolation might be something like this. If you go into these giant rift lakes in Africa or Central America, in a single lake, you might find literally hundreds of different species of what are known as cichlid fish. You can buy them in the pet store, they're gorgeous, but there are hundreds of different species of this kind of fish in one giant African rift lake.
I'm sorry, what kind of lake?
It's just called the RIFT. Okay. It's a rift, so it's just a big, very, very big lake, like Tanganyika Tanganyika is one of the larger ones, and I can't think of the others. Now, if you go into one of these lakes and you kind of study the different cichlids, what you'll find in terms of behavioral isolation is something like this. When the fish are ready to mate, the males will often build a nest. And they will defend that nest, and females will come, and if they choose to mate with that male, they'll drop their eggs, and the male will drop his sperm into the nest, and you get fertilization. The thing is that there are endless different kinds of nests and endless different mating rituals that the different species of cichlids go through. So in one species of cichlid, you might have something like a male builds kind of a volcano-like nest. It comes up and it has a little ditch on the top, and that's where they lay their eggs and sperm. In another species, they might have something flatter to the ground, and that's the nest, and the male sheds his sperm and the female drops her eggs. The thing is, and males might do a particular ritual around the volcano nest and a different ritual around the flatter nest. From the perspective of a cichlid fish, if you were a female in the species where the male builds a volcano nest, then a male who builds a flat nest might as well be an elephant in terms of the chances that you will mate with him. We have a behavioral isolation. There's something about the behavior, the nest building and the courtship, that separates out this species of cichlid from that species of cichlid. And you would be astounded at the differences in nest building and rituals that you will see in a single lake.
Is it impossible for them to crossbreed?
No.
Just choose not to.
In
time it will eventually become.
So you have hundreds of these species in the lake. I can't speak kind of categorically, but my best guess would be that if you artificially did this, most of the time, in many cases, you would be able to produce viable offspring. In many cases, you wouldn't. It sort of depends on how long evolutionarily they've been separated. Another way you can get reproductive isolation is mechanical isolation, which is just sort of a fancy way of saying, imagine a poodle and a Great Dane trying to mate. Right? The genitalia, the shape and the morphology of the genitalia won't allow it. Now, dogs are weird, so I'm not calling them different species, but they're different varieties. I just want to give you so you can picture in your mind what I mean by mechanical isolation. The genitalia it won't work. Okay. And then for plants, one way you get reproductive isolation is because they have different pollinators. And I don't talk about plants enough in my evolution lectures, so I just want to give you an example of this, and it's really a beautiful example. This is a species of Aquilegia plant, okay? And this species has relatively short, what are known as spurs, okay? And the spurs are where the nectar is, and that's what the pollinators want. And those species are primarily pollinated by bumblebees, who can kind of get in there and reach down on short spurs. There's another species in the same genus that is a closely related species of Aquilegia. I think I'm pronouncing that correctly. Right? And it has longer spurs, and it is pollinated by hummingbirds. And then there is yet another species of Aquilegia that has extraordinarily long spurs. And this is pollinated by a hawk moth that has this incredibly long proboscis that can get way down in there, get the nectar, and as it's getting the nectar, it's also pollinating because it moves from one plant, it picks up the pollen, and then it moves to another plant, and that's how pollination occurs. So here, what's the reproductive isolating mechanism between these different closely related species of Aquilegia is that they have different pollinators. Bumblebees can't get anywhere near deep enough on those. And so you don't get any cross-mating between this species and that species. And that holds true for the different combinations up there.
Could this have been because of just evolution?
The plants, so it's going to just depend on sort of where you start the story, right? The plants have these different length spurs. Bumblebees can only go into certain, and the other ones can only go into certain, and the others can go into certain, right? So what that does is it creates it creates barriers to pollination, to breeding across the species. That's an evolutionary result. That separates out those species so that we can call them species 1 of Aquilegia and species 2 and species 3. Yeah.
It seems like the moth and the hummingbird could easily access.
So the hawk moth could access the smaller one, but I don't think the hummingbird could get deep enough to get into the one that the hawk moth does. Yeah, but the longer one could
pollinate 2 and 1, and 2 could pollinate 1.
Right, but what's happened is over time, so I'm always sort of, to some extent I'm always simplifying the picture here. There's lots of other things that are going on that once you have pollinators that specialize, it sort of gets reinforced more over evolutionary time. And unfortunately, we don't have time to do an entire lecture on this thing called coevolution that looks at that exact phenomenon. Okay, so now I talked about challenges. Let me go back here. I talked about challenges for the phonetic species concept. And I said, actually, this is the species concept most evolutionary biologists use, okay? The biological species concept. But it has some problems. Okay? So again, that's the definition. And basically, if individuals in a population mate with each other but not others, then they're part of the same then they're different species. And otherwise, they're part of the same species. There are problems, though, right? And the biggest one, by far, is that 99.99% of all species that we know of are asexual. They don't reproduce sexually. They don't mate with one another in the classic sense that we're talking about here. Bacteria, viruses, some other microbial species. You can't apply this species concept. That's why when I talked about the phonetic species concept and I said it's often used in these kind of systems with bacteria and viruses and microbes, that's why, because you can't apply this system there. And then there's another issue here, and I don't want to get a lot into this because I really want to get through at least speciation after this. And that issue is just that does the breakdown of gene flow have to be absolute? In other words, does it have to be the case that individuals in population 1 never mate with individuals in population 2 for us to call them different species? What if 99% of the matings are in fact within populations and 1% is across? Does that 1% mean we're not going to call them different species? Again, subjective call, because one could make the same argument about 5% and 10% and so on. Okay, so I have to say right now, I think we should get through speciation. I don't think we're going to get to extinction, unfortunately. I want to talk about now, so now we have the sense of, okay, what is a species? And we're going to primarily rely on this biological species concept, that the breakdown of gene flow is what makes things part of one species and not another. So far, really, in the first lecture, we talked about, from the origin of species, we talked about by means of natural selection. But Darwin's real interest was in the origin of species. How do you get new species? We have millions of these things. Over evolutionary time, we've had in orders of magnitude more. How do they come about? How do you get new ones? So I'm going to talk to you about two models of how new species form, and that's known as the process of speciation, the formation of new species. The first one is called allopatric speciation. Allo comes from the root different, and patric means place. So we'll come back to that, but this is speciation that occurs in different places. The other kind of speciation we're going to look at is called sympatric speciation. Sim means same, patric still means place. This is speciation that happens in the same area. In both these models, and in fact there are other speciation models we're not going to get to, in all of them, populations evolve these reproductively isolating mechanisms. And we're going to look at how that creates new species. What differs between the two models we're going to look at is the relative geographic positions of the populations. So as speciation is occurring, is it happening in the same general area, or is it happening in two separate places? I think it'll become clearer as we walk through this. In allopatric speciation, a species splits into two when populations are geographically isolated from each other. That's how you start getting new species. When populations become geographically isolated, you build up reproductive isolating mechanisms. We'll talk about how. In sympatric speciations, populations begin to separate out into different species while they're in the same locale, same general area. So again, allopatric in different places. Let's look at this model because this is the best studied one, and it's the one that we think most often occurs in nature. And there are actually two varieties of this model. One is known as the dumbbell model, only because these things are sort of like the weights on the end of a dumbbell that you're lifting up. But the idea here is very simple. We have some large original population. Everybody's part of the same species. It's a population, a large population, everybody's part of the same species. And then some sort of geographic barrier breaks your population into two. What that geographic barrier is depends on scale. So in some cases, it might be something that the geographic barrier takes a very long time to build up. Mountain ranges can split an original population into one population on this side of the mountain and the other population on the other side. Rivers can do that as well, right? And those are long-term processes that occur. But it can happen faster, and it can happen in almost real time. So for example, today, imagine that this is a forest, and this is a clear cut. And and this species doesn't like, it lives in forests. It doesn't like to go into open, clear areas. It won't cross where the clear cut is. And if the clear cut is wide enough, you've isolated those two populations from each other. So it can be something at that level, it could be something at the level of a stream, or a river, or a mountain range, or many other things. But the key thing is the original population is broken up and into parts that are geographically isolated. Then, over evolutionary time, reproductive isolation begins to occur. And the reason it begins to occur is that the environment is never going to be exactly the same on this side of the barrier and that side of the barrier. Never. Which means that the selective pressures are going to start to act differently on this population and that population. And it might affect things like everything from what they look like to how they breed. And if it affects anything about how they choose to mate and breed, it's building up reproductive isolating mechanisms. And eventually, those reproductive isolating mechanisms become absolute. And we're now color-coding these as two different species. In other words, that barrier will at some point disappear. If it was there long enough for reproductive isolating mechanisms to evolve, then when that barrier disappears, even though there's no barrier anymore, these guys will not mate with them, and they will not mate with them. That's one variety of how you get allopatric speciation, speciation that's occurring in geographically isolated populations. This, we think, occurs, but is relatively rare compared to the other form of allopatric speciation, known as the island model. And the island model goes like this. Here's some mainland. Think of it as South America. And here's an island off over here. And in time, the things that live on the mainland may disperse over to that island. It's clear to see with birds, but it also happens with mammals and amphibians and reptiles. They can float across on wood. They colonize they colonize islands where they weren't before. If enough time passes, you're going to get, again, the buildup of reproductive isolating mechanisms between the animals here, the population here, and the population here. Living on an island is very different than living on a mainland. The selective pressures are very, very different from living on a mainland. Eventually, you may end up with a new species living on an island. Now, imagine not this simplified version where there's a mainland and an island, but the real world where there's a mainland and a dozen islands or 100 islands. And so islands are these hotbeds of speciation. They become colonized and you get the buildup of reproductive isolating mechanisms. So we think this is far more common. And again, the reason we think it's far more common is that, well, there's a number of reasons. First of all, this ecological scenario is just so much more common than this. I mean, you don't have mountain ranges doing this all that often. You don't have rivers and streams doing it all that often. But all over the world, there are mainlands and islands, many islands around the mainland. So just in terms of the number of places where it might happen, this makes it more likely, you're much more likely to see the island model in operation. But there are other more interesting reasons. And I've touched on one of them, which is that the island is going to be completely different ecologically than the mainland. Right? It's going to have a shore where all the animals, where animals are interacting with the shore much more so than on the mainland. Yes, there's a shore on the mainland, but the vast majority of animals aren't on the coast. But on the island, they're close to the shore. Island life is different. The food is different. The predators are different. Everything is different. So it generates evolutionary change. Some of that change is going to be associated with reproduction. That's going to cause reproductive isolation and speciation. That's one reason that this is such a powerful driver of speciation. Another reason is that these things aren't really to scale, right? I mean, a mainland could be much bigger than the island in terms of the scale that I'm showing you here. Okay, it could be 100 times or 1,000 times bigger than an island. What that means is that if you're looking at some species, let's say you're looking at some species of small mammal that eventually makes its way to islands. They do. They drift over on wood. They get over there one way or another. The world, the mainland that small mammal lives on is very, very large. And it's different in the northeast and the northwest and the southeast and the southwest and in the middle. If it's a very big mainland, it's just different. I mean, it's a big place. Things are different. But on the island, it's small, which means that it's across the island, things are fairly constant. That allows natural selection to operate very quickly. All the animals are essentially facing the same selective pressure. Whereas here, the selective pressures are slightly different in the northeast and the northwest and the southwest and the southeast. Mm-hmm. Yes?
I have a question. Could you also apply that to how high?
Yeah, so the question is, can you apply it? Absolutely, right? So the generic name for this is the island model, right? But this happens in valleys and mountaintops. In that sense, the mountaintop is an island on the top of the mountain, and the valley tends to be much larger area than the top of a mountain. It's the same thing, absolutely. Okay, so this really generates lots and lots of speciation. One famous example, the Galapagos finches, or what's known as Darwin's finches. Some of you will be familiar with these birds. And you find them on different islands in the Galapagos. And these are all finches. They're all finches, but they're different species. And you can see that some of them have a beak that's extraordinarily well designed for doing things like cracking open very, very tough things. And others, for example, might have a beak that's more designed for probing and that sort of thing, and across the whole spectrum. And these Darwin's finches live on different islands in the Galapagos here. And eventually, they made their way over. Eventually, an ancestral population of finches began to populate these different islands. Yes, in principle, the birds could fly from one island to another, but they don't do that very often, because even though they look kind of close in real time, in real space, they're not that close. And and because they're not, then you start building up differences across the islands, including differences in mating. And that means you start getting more and more species. We even know, based on molecular genetic information, the approximate time these different islands were colonized by finches from the mainland. And so for some of them, it was 0.03 million years ago, and some it was 1.4 million, and so on. So we know when they were colonized. We know that an ancestral population of finches did the colonizing, and now we end up with all those different species of finches. Now there you can see the key difference when you look at them is their beak morphology, but also they are reproductively isolated from each other for the most part. For the most part. And I should be clear that sometimes you can find more than one species on an island, but oftentimes you will only find one species on one island. So nature is messy, it's never perfect in terms of what we want our models to find, I should say. Okay. The other model of speciation that I want to talk about is known as sympatric, in the same place. This too requires some sort of reproductive isolating mechanism to be in place. But this is sort of a tougher nut to crack, because when populations are in different places, it's easier to see how you can get the buildup of these reproductive isolating mechanisms. But when they're in the same place, it's harder to imagine how are you going to get mechanisms that stop individuals from mating with each other if they're in the same place. So I'm going to give you, and this will probably be the last example we'll be able to do tonight, the most famous example of sympatric speciation, and this is known as the apple fly maggot, Raguletus pomonella. Okay, and right now, we know a lot about it because it's this big pest on apples, and we don't want pests on our apples, right? But there's a history here. So in 1864, this guy Benjamin Walsh, who was a friend of Charles Darwin's, was a naturalist and many other things, he noticed that there was a shift, that before this, he and others had not seen this fly on apples. What they had seen is the fly on hawthorn trees, or I guess, I don't know if I think they're considered a tree rather than a bush, but on hawthorn. Then in 1864, he began to notice populations of what we now call the apple maggot fly on both hawthorn undomesticated and undomesticated apple trees. So, not surprisingly, people have studied this species because we don't want problems with our apple crops. Now, you can find hawthorn and apple in the same fields. There is no geographical barrier that animals need to cross to go from apple trees to hawthorn bushes. But life on apple trees is different than life on hawthorn trees or bushes. Does anybody know if it's a tree or a bush? I don't know. Tree, okay, I thought it was, but I thought I once came across someone saying it was a bush. Okay, so apple trees produce fruit 3 to 4 weeks earlier. So if you are this fly, that's important. What you want to do is you want to lay your eggs when the fruit is going to be there. So there are different selective pressures for populations that are living in apple trees and those that are living, for whatever reason, in hawthorn trees. As a result, selection begins to favor different adaptations in populations that are generally living in the apple tree and those that are generally living in the hawthorn tree. And in particular, you're going to get selection for producing your offspring earlier in the season if your population is on apple trees, because that's when their fruit is. People have studied populations that appear to be specializing on apple trees versus those that appear to be specializing on hawthorn trees ever since Benjamin Walsh noticed this, but in particular over the last 50 and 60 years, when we have molecular mechanisms for determining how often mating is occurring based on gene flow. So there are all these sophisticated ways of doing this, but the key thing here is that people who study this have noticed that every generation or so, there seems to be a reduction in the number of matings between the flies that live on the apple trees and the flies that live on the hawthorn trees. So you do get some mating, but it's decreasing generation by generation. Okay? And so what evolutionary biologists would argue is that what you're seeing is what's known as incipient speciation. We're seeing the process of speciation occurring. We won't call them different species quite yet, because you still do see a significant amount of mating, but you're seeing less of it and less of it every generation as the gene flow, as mating is occurring less and less between the populations on apple trees and the ones in hawthorn bushes.
Was there some change in what was being grown in the region that precipitated this?
Oh, so that's a good question, and I don't know the answer to, did they all of a sudden start planting a lot more apple trees where Benjamin Walsh? I don't know the answer. It's a good question, though. So nowadays, we could have an entire class, but you'd fall asleep on molecular evolution. And the power that molecular evolutionary biologists have today is really kind of mind-boggling. And I just want to show you an example of the way that you can use that power to study sympatric speciation in this fly. And so I want to walk you through this experiment very briefly. So what happened was, had a bunch of researchers, right, and they went out and they grabbed flies from the populations on apple trees and flies from populations on hawthorn, and they brought them into the laboratory. Okay? And then what they did was they exposed the population that they had caught on the hawthorn tree to the world of apple trees. Okay, so they exposed the population of the hawthorn race of the apple fly maggot to conditions that mimic living on apple trees. So they created that condition in the laboratory. And then they watched to see if they saw evolutionary change over generations in real time. And the way they did it was sort of complicated, and it actually deals more with when genes are turned on and when they're turned off than whether they're there or not. But it's basically the same thing. The question was, would they see evolutionary change in real time if they took the hawthorn race and they raised them up in a world that's like an apple tree world over generations? Okay. Let me walk you through this figure. This is just a measure of the most common genes in the hawthorn race of the apple, of the fly. Okay. The red curve here is the distribution of the most common hawthorn population genes. So you see a lot of certain genes in the hawthorn population. Here's a distribution, so sort of between this 0.5 and 0.7. This curve represents the initial population that they grabbed from the hawthorn trees, and it summarizes their basic genetics. This green line here, this green curve, is measuring those same genes, but in the apple tree population when you bring them into the laboratory. So they have lots and lots of similar genes, the apple and hawthorn populations, but we're looking in the red at the most common hawthorn genes. And so if you look at those most common hawthorn genes in the apple tree version, you see that they're pushed over this way. They don't have as many of those. So this is the original hawthorn population, and this is the original apple population. Right? And what the researchers found was this blue curve is the result of mimicking the apple tree environment for the hawthorn race. And so what they saw was a movement of the genes that you typically find in the hawthorn species more towards the ones that you typically find in the apple tree species. This is hawthorn, this is apple tree, this is sort of in between. It's moved in the direction of the genes that you find more common in the apple tree. And so again, we can look at this in so many different ways. And there are other wonderful examples of sympatric speciation that we can look at. Like I said, unfortunately, I don't think we're going to be able to get through, to do extinction tonight. But I know that the slides are being posted on a slide server, and I will just leave up the slides for extinction. So if you want to take a look, and it's probably something you know a little bit about anyway, my guess is. I was going to spend a lot of time on the asteroid hypothesis for the dinosaur extinction 65 million years ago, but I guess we really don't have time for this. Next week, what we're going to do is we're going to look at the evolution of behavior, passion of my own, because that's what my lab did for 30 years. And so we'll pick up there, but now we have time for as many questions as you folks want to ask. So let's go.
How many generations is that?
Yeah, so this actually, believe it or not, is one generation. And it moved more and more over generations. But they saw this remarkable change in a single generation. Now, again, what they were measuring was basically when the genes turned on. So these were genes that were most commonly the ones that turned on in hawthorn. So they weren't actually detecting as much changes in the amount of the number of the genes as when they turned on and when they turned off. And that's why they got such a huge change in one generation. Normally, you would not expect that. And you certainly wouldn't get that kind of change in nature because this is an extraordinarily controlled environment in which they micromanage everything. Yeah.
You mentioned that the dumbbell model is less common than the island model, but even using your own example, is there like a third school of creating new species based wholly on human interaction because we're messing stuff up left and right? Yeah. So somebody was, this is a great question. So in my class at the University on Evolution over the last 2 years, I've built in a complete new kind of module on that question. So basically, we are living in what's known as the anthropogenic era, the era of where we are essentially completely changing not just the environment, but the evolutionary pressures that are operating on species. And absolutely, this is something that people are interested in and studying. And they want to know the effect of the way that we're, for the most part, screwing up the environment on how it affects not just the extinction of species, which is dramatic, but basically how they evolve, including how you can get speciation, how it affects speciation. That takes a little bit more time than some of the other things that they're studying, like how it affects the evolution of behavior, just because speciation is a process that takes a lot longer than evolution of behavior. But yes, and so just one thing before I get to the next question. Last week, there was a book over there, and apparently somebody checked it out, and it was called something like Darwin in the City or Darwin Comes to Town or something like that. I can't remember the name of it now, but that is all about evolutionary change in the Anthropocene, particularly how it affects species that are now in cities and how different an environment that is. So yes, in the back.
My question is about having the appropriate context to sift through. If your model is a certain lens, what is being left out if your bell curve could be missing the outliers that create a, so to speak, geographical expression? Because to me, there's a thermodynamic question. I was sort of curious if you ran this through really under different degrees of pressure, heating, cooling, but the whole complexities and emergence of qualities of life when the dollars that fund the research are quantitatively driven. To really understand, I think, the management, what people like to think they're doing, when really the truth of, say, Boltzmann's tomb and Bill Green, when you get into a geophysicist perspective, what is happening with ecology that they are not talking about. It's not about getting just a horse race into the game. There's a lot of stuff affecting everyone.
And there's a lot of stuff in that question, and it's a great question.
But the rate and pace of your part, I mean, even the metabolism can vary when you squeeze it down and when you just here within business management.
Right. So let me try to answer that at two different levels. First, in terms of the actual thermodynamics that you're talking about, the best answer I can give you is send me an email and I will send you the names and some papers of people that do that exact sort of thing. And the reason I'm shying away from answering that is it's just very, very far away from what I do, and I don't want to misstate what's being done on the thermodynamic approach to some of these questions. So, I'm happy to do that. I'm not trying to push off the question. And the other thing, though, that I think is at least worth touching on is what you said at the beginning about sort of what happens when you build a model and you begin to focus what you're doing through that lens and what you might be missing. So for a long time, when I first started doing this, I was involved in building mathematical models of evolutionary models. And so what I learned myself was that when people build these models, they're not trying to mimic the real world, right? That's impossible. And besides, we already have the real world. What we want to do is, and this is where it gets tricky, right? What we want to do is throw away all of the things that aren't particularly relevant and focus in on what we think from kind of first principles might be the most important phenomena. And then we build our models on that. And then we see whether or not they actually predict anything. And if they don't, then we might build in more variables into our model to see if maybe that explains explains more of what's going on. But I think that's a kind of misperception in the public, is that when you build these models, you're trying to build a model that simulates the world. What you're trying to do is get rid of all of the noise and focus in on what you think, again, from first principles, might be the most important thing.
Is that a function of the scaling in and scaling out? Essentially, just a focus.
That is absolutely part of it. Absolutely.
Yeah. I should move to some other questions too, but send me an email. Yeah.
Remember that slide of convergent evolution where there were like 3 different types? If there was a Earth-like planet, similar distance from their sun with liquid water, similar size, would we expect if there was life, would we expect creatures that would look similar to that because of that?
So this is a great question, and it's something. It's sort of I don't want to. I'm going to say it's almost a game that evolutionary biologists like to play a lot, but I don't mean to minimize it by calling it a game. I think there is a general consensus that if the environment was the same on a different planet that was, let's say like Earth, but in all characteristics, and you looked at evolution play out there, you would see at a very broad level, we would expect to see something like the process that occurred here. Now, the real catch is, like, what do you mean when you say at a very general level? What I mean is things like, yeah, I might expect that if there was water, that there might be creatures that kind of are similarly, that look very similar from very different evolutionary backgrounds that are hunting in there. But it doesn't mean that I would expect to see necessarily something that looks like a dolphin or an ichthyosaur, right? But just something that was potentially had the shape of something that lives in water. Let me again, so I try to answer these questions kind of generally here, and then I want to send you to something else, because there was this great debate that occurred on this very issue. And if you look for an argument that occurred between Stephen Jay Gould, who is a famous evolutionary biologist, so G-O-U-L-D, and a science writer by the name of Robert Wright, W-R-I-G-H-T. They got into a real thing about this question. Gould argues that things would be dramatically different if you reran evolution from the start. And Wright makes more of the argument that no one would expect humans to evolve on this planet, but we might expect parasites to evolve. We might expect predation to evolve. We might expect things at that level. And then we might expect that kind of convergent evolution. But they really get into it. So, and I think it was actually like a piece in the Atlantic or something. So it was in a magazine. Yeah. I'm going to go to people in the audience first. Yes. Yes.
Yeah, my question is, so for the ways that animals or plants will develop different species in terms of different behavioral characteristics, you gave the example of the fish with the different mating habits with the different...
Right.
And then also with the flowers, they had like different attractors to pollinators and so forth. In each of those cases, like the fish had different behavioral tendencies that caused the isolation, and the plants had a different morphology that attracted different pollinators. Where did those differences come from if they were initially kind of like a homogeneous population that was all mating the same? And so like how do populations suddenly start developing behavior preferences that are so locked in that they essentially isolate?
Yeah, so that's a great question. If you think back to the last, were you here for last time's lecture? Oh, okay. So what we talked about, one of the things we talked about was that in order for natural selection to operate, you have to have variation. So the question of how variation is generated initially is really a question largely of mutation. And mutation can occur in lots of ways, from very simple to very complex. But the truth is that if you picked any trait in almost any organism, you would see naturally occurring variation. If that variation translates into making you better suited to the environment, then selection will favor it. So the generation of the variation can be genetic. Another way that you could have variations
that variation plays an important role in, for example, speciation. One thing that we didn't talk about is think about the island model that we discussed. And imagine, like I said, that there are many, many different, I'm just actually trying to pull this up. Okay. There are many, many different islands around your mainland. One of the things that I said for the island model, let me go back to it over here, is, it's a homogeneous environment. That means selection can operate quickly. But another thing that I didn't talk about, and if we had time, we would do a series of talks on what's known as population genetics, but basically, there's something called the founder effect. And the founder effect is that initially, if we had lots and lots of islands around here being colonized from the mainland, just by chance, some of the islands might be colonized by it wouldn't be a fish in this case, but a bird or a mammal that had one behavioral variant for mating, for whatever reason, and another island was colonized by an individual from the mainland who had a slightly different, not dramatically different, maybe not enough to keep them from mating on the mainland, but when you put them on islands and they start to diverge, then they might actually turn into things that would block mating. And again, imagine that over many, many islands. So the founder effect is another thing that generates variation of a kind besides mutation. All right, so I guess, okay, so I can go back to you and then we'll stop.
Sure, my question is really about the mechanics of things not being able to reproduce widely. That's not genetic, correct?
That's not
Genetic level, correct? Like the viability of offspring, creating offspring that are viable. I'm just wondering, is there a particular thing to talk about the mechanism of changing the genome in such a way? So these two populations are, they're separated from one another geographically. There's a behavioral difference, whatever. Where is that moment where the viability of an offspring becomes possible?
Oh, okay, yeah. So, so in practice, we'll never know the answer to where is that moment in any particular system, right? All we can know oftentimes is basically what happened during the process and what we know now. And so it's a slow, generally speaking, it's going to be a relatively slow, gradual process where you might have one mating variant on this island and a different mating variant on a different island, or even in the sympatric model, you would have slightly different mating variations based, for example, on the apple tree fruit versus the hawthorn. And then what happens is, over evolutionary time, that gets magnified because you start mating more and more only with the flies in the apple trees, the more that you become specialized on apple trees and they become specialized on apple trees, and unless you mate with the ones on hawthorn bushes because they're mating later in the year because of the fruit difference. Or on the island, you're getting a slow, or even in the dumbbell model, where you have these two huge populations that are, say, separated by mountains, you get initial variation simply by splitting the population, and some individuals are here and some individuals are there, and they differ slightly. And then what happens is you get a magnification over time because selection is going to favor one behavioral variant on this side of the mountain and another behavioral variant on that side of the mountain. Why will depend on a million details of the ecology on the different sides of the mountain, but it's going to slowly build up because the selective pressure is going to be greater and greater to avoid mating with those who don't have the proper adaptations themselves, because you want to produce offspring that contain the best adaptations to that environment. Did that answer it? Okay.
Do mules fit into that story?
Do mules fit into that story? They fit in the sense that they're. Let me tell you afterwards. because I think I do want to let people go. Let me let people go. If you want to come up and talk to me for a minute, that sounds great. But thank you very much, because I know people might want to go, and I don't want to keep you.